Good evening, I'm Giulia Pericoli. I work at the Bambino Gesù Children Hospital, and today I show you my project entitled Exosome-Mediated Interclonal Interaction in Pediatric GBM and DIPG. Pediatric glioblastoma and diffuse intrinsic pontine glioma are the most aggressive tumor of the central nervous system affecting children and adolescents. These tumors are highly heterogeneous. In fact, we demonstrated that they are comprised by distinct subclones interacting in a network that maintains the malignancy of the tumor. Exosomes are small extracellular vesicles from about 30 to 150 nanometers that carry various types of molecules such as RNA, RNA, DNA, and protein. They know to mediate the cell-cell communication. Based on this, we aim to investigate the role of the exosome in mediating GBM and DIPG interclonal communication. To conduce this project, we apply multifluorescence marking technology. This technology is based on simultaneous transduction of patient-derived cell lines with 6 different lentiviral vector for the expression of different fluorescence protein. From the multicolor bark population, we obtain single-cell derived clones performing the single-cell flow sorting. Each clone has a specific optical barcode defined by the expression of the specific lentiviral vector combination. We characterize the phenotypic features such as glow, invasion, migration, and adhesion using the bright field images. On the other hand, we use the optical barcode to characterize the direct cell-cell interaction when the different subclones are in co-culture. Regarding to the analysis of indirect cell-cell interaction, exosome-mediated, we perform exosome characterization, uptake experiment, and Milne proteomic profile. As just mentioned, we apply the multifluorescence marking technologies due to the randomly different site of integration and expression strength. The 6 lentiviral vectors are integrated into each differently cells, resulting in a stable multicolor cell population, such as this GBM cell line. From the multicolor cell line, we generate single cell derived clones with a specific optical barcode. For example, 1D3 GBM clone show Katyushka, M-orange, T-sapphire, and ABFP vector, while the 5E2 clones show Venus, AGFP, and ABFP. The subclones demonstrate significantly phenotypic differences. In particular, the GBM clones show different adhesion properties on 7 different matrices. On the other hand, regarding to the DIPG clones, they show different properties regarding to morphologies and invasion pattern. In fact, the 1C5 clones show a ameboid-like invasion pattern, while the 2P4 are mesenchymal-like. These results demonstrate an intrinsic phenotypic heterogeneity that is displayed by single-cell derived clones. To study the direct cell-cell interaction, we selected the 2 most different clones for GBM cell line that have a specific and different optical barcode. In particular, we conduce the migration co-culture assay, during which we perform the single-cell tracking analysis. This analysis demonstrates that direct interclonal cell-cell interaction is a key for a more aggressive phenotype, because when the single-cell clones are together, they increase their speed and distance compared to when they are alone. Regarding to the role of the exosome in indirect cell-cell interaction, first we characterize the extracted exosome derived from the bark population and the single-cell derived clones. In particular, we characterize the protein cargo by BCA assay, and we demonstrate that we extracted the exosome by SEM and nanocyte. Furthermore, we found that GBM and DIPG subclones release exosomes, which are actively uptaken by individual clones. Analysis of the exosomal microRNAs and protein show a different profile between the 2 selective clones in each cell line. In particular, we found 3 unique Milne and 2B4 clones that are associated with the signal transduction and transcription. Regarding to 1C5 clones, we found only one unique mRNA that is associated to protein ubiquitination and negative regulation of DNA and RNA transcription. Similarly, regarding to the proteomic profile, the two clones show a different protein content. The 2b4 exosome protein cargo is associated to RNA metabolism, energy pathway, and protein metabolism, while the 1C5 protein cargo is associated with the signal transduction, cell proliferation, and communication. So we can conclude that the IPG and GBM single-cell-derived clones demonstrate significant difference when studied individually. In culture, the single-cell-derived clones display higher speed and greater distance compared to clone culture individually, suggesting that the direct cell-cell interaction is a key for their aggressive phenotype. The IPG and GBM single-cell-derived subclones secrete exosomes which are internalized and may represent a vehicle of crosstalk between the different subpopulations in the same tumor. Exosomes derived from different subclones show unique mRNA and proteomic content. Further analysis is ongoing to understand the effect of this content in the context of exosome-mediated communication between heterogeneous subclones. Thank you for attention.
So we can jump on the second speaker of the educational symposium, if it's everything, if it's all right. Philippe, we cannot hear you. Okay,
yeah, I have my microphone on. Thank you. And thank you very much to the organizer for inviting me. So today I'm going to introduce to you two imaging modalities, high-frequency imaging and photoacoustic imaging, to see the whole mouse brain in vivo and in real time. As you may know, this imaging station, the Vivo Laser X, can fit with many different application areas. So we have researchers studying cardiology, oncology, nephrology, hepatology, also doing studies on embryo development. But today I'm going to focus my talk on neurology. And some of you may be interested to know more about brain hemodynamics, brain cerebral perfusion, ischemia, thrombosis, how to detect primary tumors from secondary tumors, and how to implant biological compounds or do biopsies. Why to use ultrasound and photoacoustics? Well, these two technologies are compliant with the 3 Rs, reduce, reuse, and recycles. And these two imaging modalities are high-resolution imaging modalities. You can do in vivo and real-time imaging. These are non-ionizing techniques, so it's safe for the animal. And you can perform longitudinal studies at a very cost efficiency. More importantly, you can also extract different parameters. You can do anatomical, functional, and molecular imaging. You can extract anatomical information just looking at the tissue structure of the brain. Here, as an example, it's the rabbit pup brain. The system has different measurement tools, and it also has 3D capabilities, so you can do 2D measurements, but you can also get the 3D volume calculation done. When collecting more functional information, you can activate the color Doppler modes or power Doppler modes to look at the vessel structure into the brain, or you can even see the vascularization at a capillary level using a contrast agent. And for molecular information, you can switch to the photoacoustic mode to detect endogenous contrast, such as the hemoglobin, lipids, or metallic contents, or you can use targeted organic or inorganic contrast agents. So at Fujifilm Visual Sonics, we have a good experience working with researchers for neuroanatomy references. We have also people doing some guided injections some studying cerebral hemorrhage, ischemia, stroke models as well. The blood-brain barrier destruction detection is also a hot topic. We can detect melanoma tumors, glioblastoma tumors, study hemodynamics, inflammatory injury, and cells tracking as well. Normally, ultrasound imaging of the brain is obscured by having the skull and the skin intact. So we have incorporated brain anatomical atlas. So you simply have to scroll through the different brain slices in vivo, and then you have combined the slice from the anatomical atlas. So you can really see the region of your interest. And we use this stereotactic frame simply to keep the mouse head fixed and to be more reproducible when imaging the brain. So back to anatomical information. So here there's a very interesting work about brain PUP imaging. So here the people do an IP injection of glycerol inducing the hemorrhage. And you nicely see over this longitudinal study the location of the hemorrhage when the vessels are damaged. And this is correlated with hematoxylin eosine staining of anterior and posterior coronal section. I'm just going to select a pointer. Okay. Here we have this imaging platform. Because the animals are very small, we like to use an imaging platform where we tape the animal paws on electrodes for the monitoring of the physiological parameters, such as the animal heart rate, respiration rate, and body temperature. The ultrasound probe, the transducer, is attached to a 3D motor that you can rotate, tilt to get access to any part of the mouse anatomy. And here on the left, we have this microinjection mount where you can attach any type of needles or biopsy devices. And here, as an example, you have this pool glass needle, which is inserted into the fourth ventricle of the embryo brain. And you have another example where the quartz needle tip is lodged into the spine of the embryo. So the scale is in millimeters here. And you can see that 1 millimeter is very big on the image. So you can be very accurate when injecting viruses or any biological compounds into embryos or adult brains as well. For functional imaging, when studying cerebral hemodynamics, you can enable the color Doppler mode or the power Doppler mode in 3D. So you have a nice visualization of the main vasculature into the brain. And through this coronal section, we can nicely see the circle of Willis. And by activating the pulse wave Doppler mode, you can place the sample volume of the pulse wave Doppler into the lumen of an artery, as an example here. And you have this typical pulsative flow where you can measure the systolic velocity and end of diastolic velocity in a very accurate manner. You can also extract the mean velocity, and the system calculates indexes such as the pulsatility index and the resistive index. Another example how you can clearly visualize the main vasculature here, looking at the jugular vein, the intracranial arteries, the middle cerebral arteries, and at the background, the posterior communicating arteries. When looking at a stroke model, when the carotid artery is ligated here, you see that there's less blood flow into the ipsilateral hemisphere here. So again, it's a 3D color Doppler in rendering mode. You won't miss the stroke area here when scrolling through the different frames. So this is good when detecting arteries, capillaries down to 20 micrometers. But if you are interested in looking at very tiny capillaries that are 5 to 10 micrometers in size, then we like to use contrast agents, ultrasound compatible, consisting in microbubbles filled with gas and having a phospholipid shell. These microbubbles are made by BraYn for visual sonics. And the microbubbles are injected through an IV injection. And you can see here the ischemic area all along the skull. And we can extract using our Vivo CQ software, which is our contrast quantification software, different contrast parameters, such as the relative blood flow, the mean transit time, the time it takes for a microbubbles to loop into the image field of view, and an indication of the velocity of the microbubbles, which is the relative blood flow. And in this study, the researchers look at the area at risk. It's the region that will infarct in the absence of reperfusion. So the goal is to define this area pixel to pixel using perfusion maps to best predict the final infarct. Here we are looking at the cerebral hemodynamics in rodents. So this is the brain of a rat. The skull is open here because on the rat the skull is thicker, so you have more attenuation of the ultrasounds. But we can look at the cortex and the subcortex. And when the rat is exposed to a stimuli, you can trace the forepaws as an example. You can quantify the blood volume in different regions of interest during the neuronal stimulation. Next slide. At Fujifilm Visual Sonics, we are constantly improving and developing new features, which can be software features or hardware new features. And here we work on this super resolution microvasculature imaging software, consisting in improving the resolution on these microcapillaries. So you can extract the occurrence of the microbubbles, but you can also measure the velocity of the microbubbles and the direction of the microbubbles at a capillary level. So far, I've introduced to you what the system can do in high-frequency ultrasounds to extract some anatomical informations on functional informations. But for molecular information, you can enable the photoacoustic technology. So we have an add-on consisting connecting a laser to the system. And for those who may not know, the photoacoustic effect consists in having an optical pass and an ultrasound pass. And we have a laser, which is a pulse YAG laser, transmitted the light through these fiber optic cables, and you have this fiber optic aligned on both sides of the ultrasound window. And when the laser pulses, the light is absorbed into the tissues, and we have chromophores in the mouse body, such as the hemoglobin or the melanin, absorbing the light. And when absorbing the light, there's a thermoelastic expansion which is produced. And this thermoelastic expansion produces a pressure which produces wave sounds. And we have a co-illustration of the ultrasound image and the photoacoustic image. So you know where the photoacoustic signal comes from. So just to summarize, photoacoustic effect is light in, sound out. And one of the major application in photoacoustic mode is the detection of the endogenous contrast of the blood. So we know at which wavelengths the deoxyhemoglobin absorbs the most, and at which the oxyhemoglobin absorbs the most. And so we set the laser at 2 different wavelengths. It's a tunable laser. And then we can keep track of the oxygen saturation content. So here is an example where we ligate the coronary artery in a non-permanent manner. and then expose the mouse to a hypoxic environment, switching the gas to 7.5% of oxygen. And you see the drop in the oxygen saturation level comparing the left and the right lobe of the brain, and even more when exposing the mouse to an hypoxic environment. And when you release the notch, you see the increase of the oxygen saturation. And when back to normoxia, you see that the right lobe has not recovered as the left lobe here. And here on this movie, you see how the parametric might changes when switching the gas to a low content of oxygen. So this technique is very precise and very quick as well. So here you see in red the best oxygenated areas, and in blue the less the hypoxic areas. And the scale is in % of oxygen. This is done in 2D, but you can also do it in 3D. And this paper is a good example of following a juvenile mild traumatic brain injury. You see in 3D the oxygen content into the brain post-injury, pre-injury and post-injury. You can also see the decrease of the oxygen saturation in the 3D volume. The description of the blood-brain barrier. So you have to use a third-party device consisting in an ultrasound probe emitting high power at low frequency. So this is the way how you can disrupt the blood-brain barrier. But with the Vivo system, you can detect how efficient has been the disruption. Looking in color Doppler 3D, you can measure the diameter of the vessels. As an example here, it's the posterior cerebral artery, and you see that 2 hours post-disruption, there's a narrowing of the diameters, and then 4 days post-BBBD, the diameter recovers as well. Or you can measure the blood flow velocity as well. And here we can see the diffusion of the contrast agents after the blood-brain barrier disruptions. And this has been done at different location on the brain, and you see the photoacoustic intensity after the blood-brain barrier disruptions, showing the photoacoustic signal from the contrast agents that had been used, ICG or nanoparticles. For the detection of tumors, this lab has stated that photoacoustic allows to detect and differentiate primary tumors from secondary tumors. So here is a secondary tumor, a melanoma tumor, and melanoma tumors produce melanin, and we can detect the melanin content and see how deep is the tumor, what's the tumor shape, and we can unmix the melanin spectrum to the hemoglobin spectrum. So you can quantify the hemoglobin content to the melanin content. And when you switch to the oxyhemo mode, on the LC tissue, that's the control mouse, you see that the blood is highly oxygenated, when in the tumor tissue, the blood is less oxygenated. Some parts are hypoxic regions. And here you can quantify the hemoglobin content. So there's a lot of blood staining or having a slow motion into the tumor. And switching in color Doppler mode 3D, you see here that the vasculature is well organized, when you see some discrepancies here due to the tumor volume pressing on the vessels. So you can really extract many different parameters, just switching from one mode to another one. And this is very quick. And when looking at primary tumors, such as the glioblastoma brain tumor, we like to use a near-infrared contrast agent, which is an organic contrast agent, the Angiostamp 800. This contrast agent is commercially available, and it targets the alpha-beta-3 integrin, a cell surface receptor involved in cell adhesion and migration. And the alpha-beta-3 integrin is overexpressed in particular on endothelial cells during neoangiogenesis, as well as on many tumor cells type. This means that using these contrast agents, you can look at many different tumors, such as glioblastoma, osteosarcoma, head and neck, breast, prostate, liver, lung, ovarian cancer, as well as multiple metastasis model. So these contrast agents highly photoacoustic compatible has been validated for all these tumor types. Another example of application on the brain in photoacoustic mode is imaging inflammatory injury. And here we have the LPS stimulated the release of pro-inflammatory cytokines. And what is interesting in this paper is the fact that they report that we can measure the oxygenation content in the white matter. The system can also be used to track stem cells, as an example here. And the system can be used for in vitro studies as well as for in vivo studies. So you can look at the cells that have been labeled with the Prussian blue organic dye with nanocomplexes. And this way you can check at which concentration, minimum concentration, you can still detect the cells and how linear is the signal over the concentrations. And here we have the pre-injection photoacoustic brain image. And you can see the injection site visualizing the needle tip. And here you see the injections of the cells labeled with a Prussian dye. And then you can see how the cells are migrating into the brain tissues. So all these very interesting papers and many more are available on the Visual Sonics webpage. You can select a specific application, here as an example, neurobiology, and you can search the papers by keywords in the title and in the abstract. And this preclinical research can be translated to men. And we already have some clinical doctors using the system for clinical research. Here is the Hospital of Lund in Sweden. So they have a homemade setup allowing to do 2D and 3D imaging of the temporal artery. And the high-frequency ultrasound part has been FDA and CE approved for clinical use. So we changed the name of the system for the Vivo MD medical device. And here as well, we have an interesting paper showing what has been accomplished by visualizing the human brain using high-frequency ultrasound. Thank you very much for your attention. We have a virtual booth during the Congress. And you are very welcome to come to our booth, and through the chat window we can give you more information. So do not hesitate to come to visit us. Thank you.
Thank you very much, Philippe, for your nice talk, inspiring and very detailed.
So there's a question to Philippe Trochet. Could ultrasound and bubbles be used also to assess CSF dynamics and the possible distribution of a drug within the CSF?
Okay, sorry. Can you hear me?
Yeah,
Yes.
Yeah, okay, sorry. So I'm just saying that we can't use the microbubbles because the microbubbles have a phospholipid shell. They are very fragile. And so you can only inject the microbubbles through an IV injection. You won't be able to inject the microbubbles in another route. But to look at the CSF fluid, you can use our image-guided tool. And when injecting a dye such as the ICG, it's an organic dye, it's photoacoustic compatible, so you can switch the photoacoustic mode. And this way you would be able to visualize the fluid.
Dr. Ferrara, for the closing remarks.
Okay, no, so we just to remind you that tomorrow we will start the Congress again, 9:30. And thank you to all for listening. I hope that everything was good. Also, the scientific level was very, very high, and I'm very happy to see all of you here again. So see you tomorrow, and have a nice evening. I'm so sorry because we cannot do any social dinner or something like this. We will do the best social dinner and social party next year.
Yeah.
Thank you so much. See you tomorrow.
Bye.
Bye-bye.
Bye-bye.